The overall goal of the following experiment is to profile methyltransferases from complex protein mixtures by capture compound mass spectrometry, or CCMS. The methyltransferases are functionally isolated by trifunctional capture compounds that carry the general methyltransferase product inhibitor S-adenosyl-L-homocysteine, short SAH, as the selectivity function. In the workflow shown here, the functional isolation is achieved by incubating a cell lysate with the capture compound to establish a reversible binding equilibrium between the selectivity function of the capture compound and the target proteins. As a second step, the mixture is irradiated by UV light within the capro box. The irradiation photoactivates the reactivity function of the capture compound. This induces the formation of an irreversible covalent crosslink between the capture compound and the target proteins. Next, the capture compound protein conjugates are separated from the lysate, washed thoroughly, released from the solid support, and trypsinized in order to identify the captured proteins by mass spectrometric analysis of the tryptic peptides. Results are obtained that show the straightforward isolation and identification of methyltransferases from a cell lysate of Escherichia coli based on capture compound mass spectrometry.
Hi, I'm Lenz from the laboratory of caprotec in the Department of Biochemistry.
I'm Mathias Dreger, also from the caprotec Biochemistry laboratory.
And I'm Mirko Glinski from the Analytics department.
Today we will show you a procedure for on-beat capturing of methyltransferases from E. coli cell lysate.
We use this procedure in our laboratory to study small molecule protein interactions. The procedure enables the isolation and identification